basically today I'm going to finish a few details about B cell responses in the periphery, and then move into some kinetics information. I'll probably get more into the memory stuff on Friday, but we'll see where I end up finishing today. So last time we were talking about activation of B cells in the periphery. And specifically, we were thinking about our B cell becoming activated, undergoing clonal expansion, and then differentiating into a cell that can secrete high-level antibody as a plasma cell, potentially a memory cell, as well as doing class switching and affinity maturation. When I finished graduate school, this was kind of where we were with this. And in some ways it was like, we have the germinal center, and there was a magic wand, and that makes these things happen to our B cell. In the time since then, we have actually figured out how a lot of this works. And there is one thing that's pretty cool about this mechanism, which is where I'm going to start with on the next slide, which is that affinity maturation and class switch seem like pretty different things, right? They're very different issues. The thing they have in common is they're things that happen to B cells in a germinal center. What we have realized is that they also both require the same enzyme. And so the same enzyme actually leads to those two different effects. That enzyme is called AID. AID stands for activation-induced cytosine deaminase. And you might look at that name and say, Dr. Barker, that is a terrible acronym, because there's a C there. However, when we talked about T cell activation previously, I told you about the fact that we have that contraction phase and that cells die. That process of cell death was called activation-induced cell death, or AICD. And so in immunology, the acronym AICD was already taken when this enzyme got discovered. And so while this is an activation-induced cytosine deaminase, we call it AID, activation-induced deaminase. But I like to say the cytosine part because it helps us remember what it does. And you can see the action of AID here. So AID is an enzyme that acts on cytidines. And you can see a structure of cytidine here. So you've heard of cytidine before. What's cytidine? Cytidine is the C in DNA. C. So this is your C in DNA, okay? Activation-induced cytosine deaminase is able to modify that C base pair. So you can see it removes this amine group, in fact. So it's a deaminase because it removes the amine group. And puts this oxygen in its place. So this enzyme basically changes this chemical to this chemical. The reason why you care about this is that the product here is deoxyuridine. What's deoxyuridine? Yeah, Jamie. It's the U. It's the U in RNA, except it's actually deoxy. It's like an U, but a DNA version of it. Is there supposed to be U in DNA? No, there's not. In fact, sometimes this is where I show a meme from Chemistry Cat, where I said if I had to pick DNA or RNA, I'd pick RNA because it has U. I'm trying to be helpful here. So So this enzyme takes DNA, it specifically acts on C's in DNA and changes them to U's, which are not supposed to be in DNA. So this is sort of a problem. The difference between class switching and affinity maturation ends up being where was the C that we acted on? So what was the location of this C? And how do we deal with the U? So U in DNA is not supposed to be there. And we got to deal with it somehow. And so the how we deal with it is actually how we get to this. And so this is a figure from a different textbook. Basically, the way we do the repair either gets us somatic hypermutation in one place or one situation, or class switch in the other situation. And so this is all about dealing with that mistake of the U. So if we think about a C changing to a U, let's imagine that we don't worry about there's not supposed to be U in DNA for a second. This can lead to mutations. Why would we get a mutation, a point mutation, if we go from a C to a U? What does C base pair with? Yeah, Sebastian. G. So we've got a thing that pairs with G, and now we're going to switch it to a thing that pairs with A. And so we actually, by doing this, basically are making a point mutation in terms of what we can pair with at that location. And this kind of sort of point mutation part of it is how we have somatic hypermutation happen. So what can happen is AID can attack a U, or attack a C and turn it into a U. And we can either just not fix it and do DNA replication, in which case we get a base pair change. What used to be CG in the DNA, when we try to replicate, now becomes TA. So we've made a point mutation. Or we can do a type of repair where we just remove that one base. And in fact, we only remove the base part. We don't even mess with the backbone. We just get this little site that has no base in it. It's called an abasic site. And then we fill in a new base pair. And in fact, when you do that, you can actually fill in any base pair. So you get something random here. So what used to be a C becomes something else. You can see this in this figure from the textbook as well. We get what was a C becomes a U, thanks to AID. And then we have to deal with this open site and get a new base pair in there as part of our repair. This is specifically happening at the variable section of the heavy and light chain. And in fact, it's really happening mostly at C base pairs in the complementarity determining regions. And so at the beginning, we have relatively few of these mutations. As time goes on, and we get sort of our immune response maturing, we have more and more mutations. And you can see they're pretty much all mutations right at the C base pairs in the CDR regions. So here, AID is acting on cytosines in the CDR regions and leading to point mutations. And as a result, we get lots of mutations right here on the heavy and light chain coding genes. You can see the mutations that got picked up here. We went from no mutations to some mutations. And this changes the protein so that it has different affinity. And then, of course, that B cell can test in this germinal center, going through the germinal center reaction that we talked about. And so what I want you to get here is how this enzyme, AID, can deaminate cytosines and how that can give us somatic hypermutation. Basically, AID is making point mutations. We're kind of randomly repairing them to put in other random base pairs. And specifically, we're making point mutations at this variable region, at cytosines in the variable region. And that's letting us get these point mutations here that can eventually change the protein's affinity. So that's basically how we now understand that somatic hypermutation works. AID also can lead to class switch recombination, but the process is different. And what I want you to realize about this process is that there has to become a double-strand break. So with AID, we're basically just getting kind of point mutations, just some little point mutations. And that will give us somatic hypermutation. When we're going to class switch recombination, we have to get a double-strand break happening. And so here what will happen is we start doing some kind of repair where double-strand breaks are occurring. And this is a really sort of common example. This is sort of the easiest example to imagine, is if you were in a situation where maybe you had a bunch of C's really close together. So you didn't just have one C, you had a bunch, and so you ended up with a hole once you took out the base pairs. So imagine maybe that there was a stretch of this DNA that had like 10 C's in a row. They all got deaminated to use, and you tried to remove them. You got a bigger issue if you got a hole that's missing 10 base pairs. And that ends up looking like a double-strand break. And so the idea here is that there are multiple C's all together. We really kind of destroy that area of the DNA. And this leads us to having a double-strand break. And this will be what leads to class switching. Specifically, the area where those cytosines are in the switch regions. And so what will happen is AID will go to one of these switch regions. So with somatic hypermutation, AID was mutating the VDJ section. Here, we're mutating the switch regions. And there's a lot of C's. And so that whole switch region gets destroyed. And in order to repair that destroyed region, the cell tries to use homologous recombination of a similar sequence. So it tries to do recombination of a similar sequence to repair that destroyed DNA. And oh my gosh, there's similar sequences in front of all the constant regions. And so in fact, we'll just see homologous recombination. So to repair that process, and that will delete the intervening DNA. And so really this is actually a sort of DNA repair type process. So you can see we get these double-strand breaks, thanks to AID, just destroying the switch region. That results in these two switch regions undergoing homologous recombination so that we can get some repair. That ends up deleting the intervening DNA and giving us class switch, where we now have this new constant region exon right downstream of the VDJ. So this is kind of the general mechanism that we now understand for how somatic hypermutation and class switch occur. There's one other piece of info that we need to talk about with regard to antibody responses in the periphery. This is something known as linked recognition. Every year, this seems to throw people, and that's sad because this is so important if you actually want to use any of this information in real life. And so if you are a vaccine developer, you have to think about linked recognition in making your vaccine. If you are not a vaccine developer and you say, Dr. Barker, I am never doing this immunology business again, but I am going to do a Western blot for some other thing. And so I am going to have to make an antibody against my protein of interest. So I don't want to do any immunology, but I am going to have to immunize a mouse to get it to make me some antibodies for my Western blot. And I need to make a lot of good antibodies because I'm going to then sell them at my antibody store, make millions of dollars. Either of those situations, basically anytime you want to make a good antibody response, you need to think about linked recognition issues. And this is particularly really a problem. You kind of don't have to think about it with some kinds of antigens. But with other kinds of antigens, this becomes a huge issue. In particular, this is a problem if you are dealing with an antigen that is a carbohydrate, a lipid, nucleic acid, or a really short peptide. So a peptide that's not really like a whole long protein. If you just have like a 5 amino acid long peptide or something. In all of those situations, you have to start thinking about this process a bit. And so here we can see a situation where we are trying to make an antibody response to a polysaccharide, which is shown here as a blue flower. This other part is labeled as a toxoid. I want you to ignore the word toxoid. I'm going to call it the red part. Because that's the one thing I despise about this figure is that they call that a toxoid. Like, yes, in fact, I know why, but I just don't want to deal with toxoids right now. So we got this polysaccharide. That's a blue flower. So here's a B cell with a B cell receptor that recognizes our polysaccharide. It recognizes some epitope in this polysaccharide. And when that B cell recognizes this antigen, the B cell will internalize the antigen and degrade it, just like we saw with other aspects of thinking about the B cell. But now let's imagine this B cell wants to make IgG antibodies. So we're going to class switch to IgG, and we're going to make high level because we want to make a plasma cell. And we want to have really good somatic hypermutation. After this internalization, what has to happen? So this B cell can make all-star IgG antibodies against blue flower. What needs to happen? You can not necessarily think about looking at these details. So what has to happen to this B cell? It internalizes antigen. What's it got to do? Yeah. What? So it has endocytosed the antigen, but it has something else it needs to do after that. Yeah, Sebastian? Yes, how? Yeah, it needs to get activated by a T cell. It needs to interact with a T cell to get T cell help, right? When I told you about this process of T cell help, I told you about we would internalize the antigen, and then what will we do? Yeah.
We would go through the process of presenting our MHC.
Yeah, so we would cut up that antigen, we'd put it on MHC, and we'd show it to a T cell and be like, hello, please help me, right? So the T cell would come help. So now what if we have this polysaccharide blue flower antigen? There's a big problem. Yeah, Jamie?
Is it that the red part would also be...
Pretend there's no red part right now. Sorry. Addie? Yeah, MHC only presents proteins. MHC doesn't present polysaccharide. So we can internalize the antigen. We can internalize my blue flower. We can cut it up. But we can't put it on MHC and show it to a T cell and say, hello, please help me, right? So we're out of luck in getting this all-star B cell response. And there are lots of antibodies out there that you buy at the antibody store that are against polysaccharide antigens, or against lipid antigens, or against nucleic acid antigens, or against short peptides, all of which can't be presented on class 2. So really, when I say short peptides, I mean something too short to be presented on class 2, or something so short that it doesn't have a class 2 epitope. And you have received vaccines in your life that are giving you immune responses against polysaccharide antigen. And give you great, beautiful immune responses. I believe your diphtheria, I think actually both your pertussis and your diphtheria are polysaccharide. You probably have beautiful antibodies against them. But based on what I've told you so far, that's not possible, right? We got a flaw in this plan. So the way that we actually deal with this is what's shown here, which is that we take a protein. Again, we don't have to call it a toxoid. We'll just call it a protein. It's a little red thing. And we actually physically attach it to our polysaccharide antigen. Oftentimes, we call the polysaccharide, or whatever the antigen is, we often call it a hapten. And the protein that we're attaching to help out is called the carrier. There are a bunch of proteins immunologists like to use as carriers. The one that I often think about is called KLH, one called CGG. There's BSA sometimes. There's a whole big long list. I often think about KLH, which is called keyhole limpet hemocyanin. It's this weird, big protein that's super repetitive and has some really nice class 2 epitopes. And somebody like in 1900 tried it for one of these experiments and it worked, and it's just like the one people use. It comes from an organism called a limpet, keyhole limpet hemocyanin. And a limpet is a super weird organism that is very odd, and I don't even like to look at. It kind of looks like a tongue. So we'll take one of these carrier proteins and we'll attach it to our polysaccharide. And one thing that's super important here is that the carrier has to be physically attached to the polysaccharide. You can't say, put one syringe in with your polysaccharide and another syringe with your carrier, like at different times. You can't even just have them both in the same syringe. They have to be physically, biochemically attached to each other. In fact, they must be linked for linked recognition to work. And when that happens, the B cell will bind to that polysaccharide with its specific BCR. It will internalize both the polysaccharide and the carrier. And it will degrade this polysaccharide and carrier. If they were not physically bound together, they would not get internalized by the same B cell. If they were not actually like physically bound together, one B cell would internalize the blue part and one would internalize the red part. You need a B cell to internalize both of them. So they have to be physically linked. And they both get degraded. Then the MHC can actually present part of that red carrier protein. Now suddenly you have an epitope to present on class 2 and activate a T cell so that the T cell can actually provide help. And so you can see the T cell is actually not responding to the same epitope as the B cell at all. But because you physically linked them together, you can get this T cell to help out this B cell. And so you'll get a limpet-specific T cell that's going to help out this polysaccharide B cell. You're kind of taking advantage of your immunology knowledge. That will help the B cell. That'll let the B cell class switch, become a plasma cell, do all that good stuff, and to make your lovely antibodies against your polysaccharide. And you can see this in this figure from your textbook as well. So usually what you'll do is you'll take your little Hapten, which here is red triangle. They're calling it DNP. And if you just inject that Hapten by itself, you get no antibodies because there's no possibility of T cell help. You can also inject just your carrier protein. In this case, they have BSA. You will get some lovely antibodies to BSA. Great. No one cares. Not useful. Not what you're going to make millions of dollars at your antibody company with. But if you actually physically bind the two together and immunize an animal, you'll get some antibodies to the Hapten alone, some antibodies to the carrier alone, and even sometimes antibodies to like the bridge point of them. And then you can actually do some additional work to sort out the B cells that just make these antibodies and purify those B cells. So you will have to do some additional work on sorting the B cells, but this is how you can get those antibodies. And when you got immunized with those vaccines in your life that were for polysaccharide antigen, they were all attached to a particular carrier protein. You didn't just get the polysaccharide alone. Yeah, Jamie? Okay.
So when the B cell phagocytoses the polysaccharide with the carrier attached to it, it then cuts that up, and the thing that gets presented on MHC is the carrier.
Is a peptide from the carrier, yes.
So then how does it end up making a response to the polysaccharide?
Let's see. Okay, so this B cell is specific to the polysaccharide, okay? So remember, every B cell only has one specificity. So this B cell is specific to the polysaccharide. That's how it did the internalizing. That same B cell, once it has internalized, will present. So here you can see it's the one with the polysaccharide, it internalized. And the T cell doesn't care, doesn't like check to see that these match. The T cell just says, oh, you are presenting antigen. I'm going to help you. And so as long as we have some way that the T cell can see an antigen and say, okay, I'm going to help you, it doesn't actually matter if they match. Okay? Does that make sense? And so the point is, the reason why this B cell is specific and the reason why you get antigen to the polysaccharide is because the original B cell internalized the polysaccharide. It had the B cell receptor for doing so. Okay? Yes? No? Okay. This is like super important for actually using this info. All right, so now I want to talk a little bit about aspects of immune response kinetics and phases of what we know about kind of kinetics of the responses. And so this is one of many kind of general schemes to show you the kinetics of an adaptive immune response. So you can see our naive B cell and T cell. Note that they were present before the antigen was ever there. So they were made by VDJ recombination in the absence of antigen. They're waiting around for that antigen to happen. If the antigen is there, either presented by an APC in the case of a T cell or as free antigen in the case of a B cell, we're going to have clonal expansion and get many copies of that B cell or T cell. That might make your lymph node swell because this is happening in your secondary lymphoid organ. Those cells are going to differentiate, and we're going to talk a little bit more about differentiation of those cells in a bit. But the differentiation includes things like going from being a CD8 T cell to a CTL, or going from being a TH0 cell to becoming a special kind of helper T cell making cytokine, or going from a plain old B cell to a plasma cell who has class switched and who has done affinity maturation. We may have to have those cells go to the location where they need to go in order to get rid of microbes. And then we're going to have the contraction phase, where we're going to have some of those cells decrease in number. And some of those cells are also going to be surviving memory cells, which have some new properties. Okay? And so you can kind of see this general overview. This is what it should look like. So I'm going to flip the order of this because I feel like it. And so what you can see is that eventually our cells are all going to go from naive cells to effector cells. That's the sort of broad overview of what we've talked about. And we can think a little bit about the difference between naive cells and effector cells. So you can see some of this here. So this is a table from a textbook. This is sort of this idea of this for T cells. This is a T cell situation. I'll also show you the B cell situation on the next slide, and we'll kind of talk about effector cells in general. One big difference between naive cells and effector cells is that they are making way more of some of our really important proteins for the T cell. So you can see, for example, with IL-2, an effector T cell makes 1,000 times as much IL-2 as a naive T cell. 100 times as much of that transcription factor FOS. 100 times more of many of these other cytokines. More cell cycle proteins like cyclins. There are more proteins that are involved in cell survival. More proteins that are involved in cell division. All that good stuff. So that's pretty cool, right? The cell is highly specialized. The cell also is going to do one other thing. And I'm realizing, when I was editing these slides last night, there was one slide that I was going back and forth on whether I wanted it. And I was like, no, I guess I don't need that. And I want it right now. So that's fabulous. But these cells are also changing their trafficking proteins on their cell surface. So now our effector T cell is not going to have proteins on its surface that make it go to the lymph node. No more L-selectin. No more CCR7. Instead, that cell is going to have some kind of trafficking proteins that send it to a tissue. If it was, say, a SARS-CoV-2 specific T cell, it might now have the right trafficking proteins to go to the lung. So it loses the trafficking proteins to go to the lymph node, and it gains the ones to go to the lung. That's kind of important because remember I told you with the CD8 T cell, it differentiates in the lymph node. It originally couldn't kill dendritic cells, and then it gains the ability to kill. If it stayed in the lymph node, then it would just kill the dendritic cell. But it doesn't. It leaves and goes where the virus is and goes and kills virus-infected cells. And so it goes to where it's going to be useful instead of just chilling in the lymph node. So, if you have a viral infection of the skin, the T cells are going to go to the skin and kill it, kill that virus. So there's also a big trafficking change in terms of effector cells trafficking to different locations. The effector cell is really similar with the B cell in terms of it being a plasma cell. And you can see, again, the very highly specialized plasma cell here, and how specialized that plasma cell is in that it's basically just ER so that we can secrete. The cell secretes antibody and pretty much does nothing else. We often talk about those plasma cells also going to the bone marrow and living in bone marrow. You can sort of imagine it being a little less important where a B cell lives because it's secreting stuff into the blood and the antibodies can go around the whole body in the blood. It's not like the B cell has to be in the lung, although we're realizing now a bunch of them go there or go to wherever the site is. So the classic definition was like, oh, we don't care where that B cell goes. We kind of do now. If my sister was here, she'd be very angry with me for not caring about where the B cell goes. And so this is kind of the idea of what that response looks like. So you can see we have our nice primary response, and that primary response is going to get rid of the virus. But there are a couple things I want to sort of tell you about to sort of apply this info. So you've seen this slide from me a few times now. We feel good about this? We feel like this makes sense? This information, you can see in the immunology book I took this from, it's chapter 1, is clearly not understood by certain members of the press right now. And there is a lot of argument going on about some of this information. And this is one thing that has been really interesting in the past couple of years and during sort of the SARS-CoV-2 pandemic, is that this is a pandemic where we are actually able to really measure immune responses in real time during a pandemic, and with a whole bunch of really fancy techniques that we didn't always have before. And so we have been able to measure what happens to immune responses in people who are infected with this virus or who are vaccinated. I think all but one of my examples are infection examples, not vaccination examples. And we've been able to measure things in real time. And in most cases, you know what we see? We see pretty much some things that pretty much exactly correspond with this. Yet, it's some of the first times we've had really beautiful data, like making it so obvious. And so we keep kind of reproving immunology is true throughout this. But we've seen some, but it's really interesting to actually see what comes out. And so I want to show you a little bit of how this looks in the real world with SARS-CoV-2. And so again, remember this curve. So one of the big questions and things that has been debated in thinking about immune responses to SARS-CoV-2 after infection is, how long does the immune response last? I always crack up at these. These basically were published like about the same day. They were very close together published papers. In both cases, they were measuring antibody levels with slightly different ways, days after symptom onset. And so there's two papers looking at antibody levels, days. You can see sort of what happens to the antibody levels in days after symptoms here. This paper is called Robust Antibodies for Months. This paper is called Decline of Antibodies in Months. Let me look at the data. Do these two papers find different things? Nope. It's just what they call them, and if they call them robust or decline, right? But now let's think about what we know about immune responses. What should happen to your antibody responses over time? Yeah. They should wane. You should actually stop having those effector cells producing lots of antibodies and going down over time. That's how it's supposed to work. In fact, a piece that I'm now remembering I forgot to tell you about effector cells is that effector cells are usually thought of as being extremely short-lived cells, or relatively short-lived cells relative to the other T cells and B cells. They live for a relatively short time. And you can kind of imagine this when you're thinking about the plasma cell. Look at how much specialization this plasma cell has undergone. Does this plasma cell have like a lot of stuff to do cell cycle? No. This plasma cell seems to have a lot of good ways to survive. This plasma cell is basically using all of its energy for this one purpose, and then it's going to die. And so it's going to make you tons of antibody, and then it's done. And so typically, and this is also true with our effector T cells, we tend to think of them as being somewhat shorter lived, again, compared to the other T cells and B cells. In fact, some people in the field call them SLEX, or short-lived effector cells. So our effector cell is going to be short-lived. We want it to be short-lived because we don't want to become a walking lymph node. We need room for our next effector cells the next time we get infected. So we should have a decline in immune responses. And so everyone who's freaking out like, oh my gosh, the antibodies are going away, maybe freaking out over nothing. What you really want to know with these kind of data is this decline more or less than what it should be? Which is something that people are still trying to figure out. But there are some other things that have been figured out. This is one of a bunch of papers that have done this. I will tell you, this paper actually came out pretty recently. I have white boxes over some of their data because this paper actually makes a bunch of really fabulous points. And I don't want to get us confused by going on one of the other points. So I'm just trying to show you the data that is on the point that I wanted to do. But this is actually looking at SARS-CoV-2 antibodies following infection. And so here they are looking at antibodies against 2 different antigens at people 1.3, 6.2, or 12 months after infection. And so if you look at this, what do you see? Yeah, Rishi. It's not significantly different between 6.2 and 12 in this case. There are some that are significant, however. But what's the overall thing, Sebastian? Yes, so the whole thing differs between different antigens. But in general, there seem to be fewer antibodies a year after infection than there is at a month after infection. Okay, so now what's the New York Times say from these data? Huh? You lose so many antibodies. You are less protected a year after infection. Disaster! It's terrible. You don't want to be in this boat, right? According to this, 12 months, bad. Right? Yeah? But then let's look at some other data here. These data show you the somatic hypermutation, the number of mutations that the B cells making those antibodies have. So remember, somatic hypermutation is going to give you better and better quality antibodies, antibodies that bind better. So this is showing you somatic hypermutation of both the heavy chain and the light chain. What do you notice about the somatic hypermutation? This did have statistics on it. It was so confusing, I covered that up too. Yeah. Yeah, you have fewer antibodies, but they're better. Their quality's way better. And this is actually basically a binding strength. So, or it's getting at how quickly the antibodies dissociate, or how much it's effective concentrations, how much antibody you need to inhibit. Lower is better here. Means you need less antibodies to have the same activity. Lower effective concentration means your antibodies are better, stronger antibodies. So what do you see from these data? They're getting stronger. So you got more somatic hypermutation and stronger antibodies over time. So it's not just about how many antibodies you have. This isn't just a quantity issue. This is also a quality issue. And you can see quality goes way up. And you can actually see that there is quite a bit of a difference between 6 months and 12 months after infection. Like, you got to wait a while to get the really good ones. Because you got to wait a while to let somatic hypermutation and the whole germinal center reaction thing happen, right? And so now if you look at these data compared to, and you look at both this and the data I showed you on the previous slide, would you rather have 6 months antibodies or 12 months antibodies? Which one would you rather have? 12 months antibodies. Yeah, there's fewer of them, but they're way better. These data are a little bit tricky to look at. I'm going to try to walk you through. Try to walk you through these data. So here they're looking at pairs of antibodies from different patients. And they're showing, basically, they're showing you whether or how much antibody is required to act against different versions of the virus. So this is saying, do the antibodies work against variants? And in each pair, they've got one from 1 month and one from 12 months. And so and in red means basically bad and useless. White means they work. And so you can see that, like, here's a situation. In some cases, the antibodies at 1 month are not good against variants. But at 12 months, fine. And you can see this in many examples here. This is actually telling you at 1 month and 12 months what percent will act at different levels. So here you can see that you've got some relatively weak ones, some kind of medium ones. And if you look at the variants, you've got some that like don't work at all against the variants. But at 12 months, a lot of that red, the ones that don't work at all, are gone. So in fact, not only have you increased binding strength and effect, you've also increased breadth and how broadly your antibodies can act after 12 months. And as they say, in conclusion, continued clonal evolution of the antibodies over 12 months favors increasing potency and breadth, resulting in antibodies with an exceptional activity against a wide group of variants. Yeah, if you look at your antibodies 5 minutes after vaccination, they're not going to be very broad or very good. All of the data that looks long-term after any of this says that your immune system does not care about variants. And what you can see is how much your immune response is actually improving over time. And so there's a big change in quality through this activation part of the process, and through, in fact, some of these cells changing into memory cells. What you should realize here is that whenever somebody says the immune response does X, the immune response means a lot of different things. Do they mean quantity? Do they mean quality? Do they mean breadth? Do they mean innate? Do they mean adaptive? Do they mean T cells? Do they mean B cells? And so there's so many times where people will be like, this is what happens to the immune system. And I'll be like, oh, that's cute. Because, again, you should realize that there is, in fact, a lot more distinction. And so you can see from those data that we have actually been able to show with SARS-CoV-2 infection just exactly how nicely this process works and how much of a change you get over time, and how, in fact, to get the best quality B cells, you need to let that germinal center reaction that we talked about before have some time to do its thing. You don't necessarily want to think about what's your antibody level look like right after infection. You actually need to give those B cells time to go through their full differentiation process. Yep, Sebastian.
If our antibodies are getting better over time, then how come we need the flu shot every year?
Good question. So the reason why you need a flu shot every year is that the influenza virus has special mechanisms of genetic change. And so the virus each year is actually different. And so each year you are getting a flu shot against this year's virus. You don't have that happen with most other viruses because they don't have the same types of genetic change. And there are efforts underway to change the way the flu virus is made so that the genetic change doesn't matter, but we have not yet succeeded at that. So it's about genetic change in the flu case, not about the immune kinetics. All right, so I guess one other thing that I will point out, and again, this is something that I will mention. Where did I put the slide where I mentioned this? No, a lot later. Okay. Sure, fine. And so here you can kind of see this response, both our primary response that we've been talking about, and then the fact that later on, if you are infected again, that you do have this different type of response known as a secondary response. And that's because, again, of the changes that have happened to the cell and the sort of changes in the cell's quality. There is a change in quantity. You can see we go from like not really measurable to a little bit measurable. So there is a change in quantity, but there's such a big change in quality that we see something very different later on. Also, I want you to notice on this slide, and I actually was using this image very much on purpose, that when we are talking about that second or later infection, we are talking about a later infection that is happening much later. We're not talking about an infection that happens like a month later. We're talking about something that happens way, way, way later. And the reason is that we need those cells to get this response that I'm showing you to have undergone enough differentiation to be able to make this changed quality of response. If you get infected here, where those cells have not fully differentiated, yeah, you're still going to have lots of plasma cells or TTLs around, and that'll probably just kill the pathogen, and that won't even like count. Here you can see it's called an abortive infection. And so those aren't really things that are going to push you to this nice secondary response. You've got to actually wait a little while. You've got to have some time for those cells to change their quality so that they can make this nice response. And so this response is mediated by a type of cell called a memory cell. I don't know where's the thing I want. I'm going to go with this one. And so you can see that at the end of our whole primary response process, we end up with cells that have these new capabilities called memory cells. And so in order to get that lovely secondary response, we need to make sure the cells get enough time to differentiate into memory cells. So I'm just going to go with the weird order that I want in my head right now. Memory cells do have a lot of differences compared to either naive cells or effector cells. But I want you to think for a second, or I guess I'll let you think for a second, but then I'll just tell you the answer, about this and one application of this piece of information, because this is another thing that has been like the debate of the past couple of years in immunology circles. There's this question right now being discussed all the time about boosters. You need a booster vaccine, right? And I will tell you that from looking at data and from my understanding of things, my answer to that has evolved a bit over time in terms of how I feel about boosters and immune responses to boosters. And one piece of that is actually very much related to this. So if you were to get the Pfizer vaccine for SARS-CoV-2, what is the Pfizer vaccine regimen right now? So just the regular, not the booster. Yeah.
So you'd get the first shot and then 3 months later you'd get the second?
Yep, so Pfizer. Right? If you do a booster, Okay, what's the deal with Moderna? Dose 1 is at day 0. That's easy. Do you guys know? Yeah, Krishna? Yeah. So dose 2 in this case is day 28. And they're also boosting at 6 months. So let's imagine we don't have a booster first. Let's look at this information. Can you see any potential problems if we don't think about the booster? Yeah, Sebastian.
Dose 2 would be part of the aborted infection.
Yeah. Is that second dose too close to the first dose to give you the most perfect memory response? I mean, you're still going to get some memory response. You're still going to get some nice response. But the sort of classic idea is that you have to wait longer before you get the perfect memory response. And maybe if you wait longer, you're going to have better quality of those cells and make a better memory response. Does that make sense? Why is it that Pfizer and Moderna went with those kind of times and didn't say, oh, we think that the best possible immune response happens if you get your second shot at 3 months? Why did they go with a month or less instead of 3 months? Yeah, Rishi. Yeah, they wanted you to get 2 shots as quick as you could, so you were protected. And so they're like, maybe that is not the most perfect, ideal, gorgeousest. Is that a word, gorgeousest? Definitely not. Okay, perfect. I mean, maybe that doesn't give you the best possible immune response, but it gives you one that's good enough and gets you back out into the world as fast as possible and gets you ready to combat this virus as fast as possible. So that's why they went with those times. And that is true. That is why they went with those times. But we have had some more time now. Where people have actually been able to look at the difference between a short interval and a long interval. What happens if you actually do the boost later? And the difference is actually, so they delay the boost for about 10 weeks. It's actually between 6 and 14, because this was like in real people. They were studying what really happened with people. Look at the antibody response, short versus long. You can see that that long interval actually does give you a better antibody response. This is, especially with the CD4 response, and the gray is more important here than the red. You can also see the T cell response is actually better with the long interval if you do allow those cells more time to really differentiate into memory cells. And so that is kind of part of this timing. And the piece of data that I have found the most convincing for boosts, and again, like I said, I'm sort of on the fence on them, but the piece of data that to me is the most convincing is the information that says, yeah, when you get to a boost, you're actually here. You're actually at this place where you have the good memory responses and you can make the pretty immunology graph. You're not in this abortive infection phase anymore. You're really, you finally waited long enough for real nice pretty memory cells that can really give you a good secondary response. And so, again, there's data for and against boosting, and I have feelings on both sides of it. But this, to me, is the most compelling reason, is because we actually want to wait for that immune response quality to change and for us to have the good memory cells that can do memory cell things. And on Friday, we're going to talk about what those memory cell things are and the specifics of memory cells. And we're also going to go through some cool applications of all of this info.